Second talk might be delivered by the Dr. Lee, Hae-Ock Lee, and she's in the her institute is Samsung Genome Institute, and she run the many work regarding the single-cell RNA sequencing. And her talk title is Characterization of the Tissue Remodeling and Aberrant Anti-Cancer Immunity during the Lung Cancer Progression. And give a big hand to Dr. Ray.
Thank you. I'll be talking about the lung adenocarcinoma progression today using single-cell RNA sequencing data. I have used the 10x genomics data, so it's a large number of cells and shallow sequencing. We have sequenced about 20,000 reads per cell on average. As you all know, lung cancer is the leading cause of cancer cell deaths in Korea and worldwide. And let me remind you that over 70% of the lung cancer is diagnosed at metastatic stage. So it's really important to understand not only the primary tumor, but also the metastatic disease. And of the stage 4 lung cancer patient, about 40% of them go to the brain. So it's considered the final stage of the lung cancer, and the survival expectancy is very low. So it's really important to understand those metastatic disease. There have been many targeted drugs introduced to lung cancer. It's actually very famous cancer for the molecular targeted therapy, for example, the EGFR, and also for immunotherapy. So it's targeting actually both cancer and immune microenvironment. So it's, again, very important to understand not only the cancer cells and also the microenvironment, including the immune system. There have been many cell types that is involved in the tumor progression as well as the therapeutic success, especially immunotherapy. For example, the tumor-associated macrophages usually considered to be anti-inflammatory and bad for the progression. Regulatory T cells are also considered a bad population to suppress the immune system. Fibroblasts in general are considered to be pro-tumorigenic, but the involvement of the fibroblasts in lung cancer is not as important as the colon cancer. So there are many different cell populations affecting the progression of the lung cancer. This year, there have been 2 papers came out using the single-cell data to look at different population in lung cancer. One is by Jamin Zhang's group, who have sorted T cells using different markers like a CD4, CD8, CD25 to target different T cell population, and use the transcriptome data to cluster different T cell populations. And these are the number of clusters they have generated, and found some important, potentially important clusters like a CD8 LAYN population, which is a more exhausted CD8 T cell population, and there's also a more effector-like T cell population. In another study by Louvain group in Belgium, they have looked at the lung cancer from the primary site. Actually, this study was also primary site, and they have looked at the peripheral blood cells as well for the comparison of the different T cells. For this study, this group have looked at all the cells they could look at by dissociating the tumor tissues, and it included 3 adenocarcinoma patients and some different subtypes as well. So both has its own advantage and disadvantage. This is very good to look at the T cells with deep sequencing, and this one, it's a shallow sequencing for each cell types, but you can look at the relationship of the different cell types. So we adopted the second strategy to look at different population. and we focused on more on the metastatic disease. So this is our cohort, 58 samples from 43 lung adenocarcinoma patient. We have focused on only the adenocarcinoma patient, the most prevalent subtype of the lung cancer. We actually aimed at looking at stage 4 disease with the biopsy samples, but then we realized that many lung cancer biopsies are from the lymph node. Some are from tissues. So we had a need to look at the not only the matched normal tissues for the comparison, but also for the lymph node, because without the tumor infiltration or tumor metastasis, they have very different microenvironment and very different resident cells. So comparing, let's say, these two tumors only to the lung tissues may give a very different or sometimes a wrongful interpretation of the data. So we had these biopsy samples from lymph node and tissues. Some we had matched fluoroeffusion samples. This is different patient, 10 brain metastasis samples, and as a control, and also a very good initial study material, they matched primary tumor and distant normal tissues. We also had lymph node from the lung cancer patient, which is non-metastasized. This is the landscape we have drawn from these patients. So we have looked at over 200,000 single cells from the primary and metastatic sites. With the SORA algorithms, we have identified 9 major cell types. We have used the different algorithms and also different parameters to make the clusters, and usually it was 9 major cell types. So all major cell types were contributed by different samples. There are some sample-specific prevalence in certain type of cells, but it was not patient-specific. This is a brief overview of the data processing. As you may all know, the genomic data, the transcriptomic data, you need to have the transcript count. So in this data set, you need a cell barcode to see which cells gave certain transcripts. So that's the difference of this data compared to the conventional transcriptome analysis. And of course, the data has many zeros, like cells with very few gene expression. You have to remove them, otherwise it's gonna mess up your analysis. So there's a heavy filtration steps, and after using all the filtration, we had about between 2,000 to 3,000 cells from each tissues or each sample, and about 1,000 to 2,000 genes per cell. And this is again brief analysis overview. We have done different analysis, but most of the analysis starts from the clustering and identification of the clusters, which cell type they are. So we have used the single cell transcriptome data to define the cell types and compare them between the different tumor tissues and also different patients. So the first landscape that I showed you, we have aggregated all the data. So that was from different tissues, and this data shows those cells within the origin. So this is from the normal lung, the primary tumor, the biopsies from both the lymph node and tissues, fluoroeffusion brain site, metastatic brain. This is a lymph node without tumor cells. So these two data are the biopsy samples from either the lymph node or tissues. And as you can see, actually, they show some different profile of the cells. It's partly because they are from the different site. One's from the tissues and lymph node. But if you compare the normal lymph node and metastatic lymph node, there's also differences in the cell types, which is a contribution from the tumor. So actually, there's a combination of this, the original microenvironment and also new environment made by tumor cells. This is a brief cell type proportion that we have found in different tissues. The red cells are the epithelial cells. So for normal, it would be normal epithelial cells, and tumor, it would be mostly tumor cells. Again, the data shows the different proportions of the epithelial cells. The number of tumor cells actually from the tumor burden, but also from the characteristics of the tissues. So it's not directly correlate with the tumor burden. So when we compare the proportion of different cell types, we focused on single cell type. For example, here, these are all immune cells. So if we compare the cellular proportion, including the epithelial cells, it would actually distort our data. So we have only compared like a similar cells of the similar characteristics. So if you look at the summary data here, compared to the normal lung, there is a slight increase in T cells, decrease in myeloid cells, and large increase in B cell compartment. So this proportional changes were consistent in different tissues. After we looked at the cellular proportion, we tore apart the different cell types and started to look at the characteristics of each cell type. This one shows the epithelial cells in normal lung. So this is more of the side data we have got. So we have looked at, we found 4 cell types, the alveolar type 1-2 cells, club cells, and ciliated cells, which have been reported before. And we used a similar clustering strategy to look at the cancer cells. And this is that they make all patient-specific cluster. Itae has mentioned this before, that each patient has very different genetic aberrations, and each cancer cells have their unique gene expression features. So with this type of analysis, the unsupervised analysis, it was not possible to make a grouping of tumor types, like 3, 4, 5 molecular subtypes. We wanted to devise some certain common themes, common characteristics to group the cancer cells, but the unsupervised clustering, it was not really possible. So we started exploring different methods. And one method is it's a different clustering method. This is a trajectory analysis. Using this one, actually, we were able to classify our tumor cells into 3 groups. And one group, so there was 3 groups of cancer cells. One was there was less cancer cells. The majority of the cancer cells were either the stage 2 or stage 1 group. Stage 2 group had common differentiation related genes with the normal lung tissues, and stage 2 cells actually had very different gene expression data. And when we looked at each patient has either stage 1 specific cells, so majority of the cells were stage 1 specific, or the other groups were stage 2 specific. So there's always a mixture of cells, but there's a more stage 2 enriched patient or stage 1 enriched patient. The number of patients for the primary lung cancer, it's only about 10, so it's not possible to make a statistic analysis. But the stage 2 was, there were more advanced stage cancer and related more with the smoking status. So to look at the importance of this stage 1 or 2 of the cancer cells, we looked at the other data set, the bulk expression data set from TCGA, which has the patient survival data, and used the gene sets from either from stage 1 and stage 2, and we found the stage 2 actually, the patient with the higher stage 2 expression has a worse prognosis. And this pattern could be seen in our single cell data. So for the primary lung cancer, there are 2 groups of patients. more stage 2 ones with the more stage 2 cells, or ones with more stage 1 cells. And in the metastatic site, the signature was elevated. The stage 2 signature was elevated. So we concluded that this stage 2 specific signature is associated tumor progression and metastasis. So after looking at the cancer cells and you have done the molecular subtyping, we moved on to tumor microenvironment. So this data shows the fibroblast data. So this is the power of this single cell analysis. With single data, you can look at all different population. From the fibroblast analysis, we were able to identify over 10 fibroblast clusters, and we have named cancer-associated fibroblasts for the clusters with more tumor-enriched sample. So in each type of fibroblast, for example, the COL13A1 matrix type of fibroblast, there's a matching CAF, the cancer-associated fibroblast. It was all the same for other types of matrix fibroblasts, myofibroblast, and others. And when we looked at the gene expression signature here, so this alpha SMA is a famous CAF markers, as well as the myofibroblast marker. You can see here that actually the CAF marker, the alpha SMA as a CAF marker, actually stands for the expansion of the myofibroblast, whereas the FAP, another CAF-associated marker, it actually marks the transition of different type of fibroblast into the activated state. And since we gathered different information from different cell populations, we have looked at the relationship of the fibroblast and T cells here. So the T cells, especially regulatory T cells, are recruited to the tumor site or any immune site with the CXCL12 chemokines, and we found that the matrix fibroblasts are the major sources of CXCL12, and there's a correlation between the higher proportion of CXCL12 positive matrix fibroblasts with a proportion of the regulatory T cells. So this data shows that actually fibroblasts are the initial driving force recruiting the regulatory T cell population into the tumor immune microenvironment. And using the different CAFs, we were able to delineate the CAF-inducing factors. So this time we have used the intersect gene sets for different type of fibroblasts, and we could list 27 common genes that might be a common CAF-inducing factors. And this shows the dynamic changes of fibroblast proportion during lung cancer progression. As you see here, in the normal lung tissues, it's mostly matrix fibroblast, and this proportion changes to more myofibroblast in the primary lung cancer. And in the metastatic site or brain, there are very few fibroblasts, but it's mostly the myofibroblast. And they are known to play a critical role in making tumor microenvironments. So they would be the target if we want to change the tumor microenvironment. The B cell part, actually the most dramatic change was the large increase in the B cell proportions. In the normal lung tissues, there are very few B cells present, B cells and plasma cells present. However, in the lung cancer site, the number of both B cells and plasma cells increases. And it was actually for most of the isotypes. So this data shows that whether it works or not, there is an activation of humoral immune response in the lung cancer site. Macrophages are the huge population taking up the lung tissues. And it's known that in most of the cancer, macrophages tend to have an anti-inflammatory phenotype, which may not be true for a certain type of cancer. For example, the colon, they may have more inflammatory signatures. But in lung cancer, we were able to see that compared to the normal tissues, the macrophages from the tumor tissues actually have the anti-inflammatory gene expression. So this one shows the macrophages in the metastatic lymph nodes. If you know about the normal lymph node, you don't find the macrophages there. So this is cancer-driven changes in the immune microenvironment of the metastatic lymph node. And actually, most of them were anti-inflammatory type, but there are a few with more pro-inflammatory signature. This pro-inflammatory macrophages had gene expression signatures indicating they are recent immigrant from the blood. So they may be like a bone marrow-derived suppressor cells or something like that. Similar findings in the tissues. So most of the pro-inflammatory macrophages were from the normal lung tissues. So there's a lot of alveolar macrophages. So these are the normal resident of the lung. Compared to that, the tumor-associated macrophages had more anti-inflammatory gene expression, though they actually demonstrated quite a difference. One more with the tissue-resident macrophage markers, the other more blood-derived macrophage markers. So it seems like the increase in the macrophage in tumor microenvironment happens through the expansion of the local macrophages and also recruitment of the blood or bone marrow. The last part of the cell population that I'd like to talk about is the T cells and NK cells. We have analyzed both populations together because in a transcriptome way, actually they share very similar gene expression signatures. So without any sorting strategy, it wasn't really possible to look at them separately. Anyway, here we have looked at over 15 different cell populations. Largely, there were CD4, CD8 cells, NK cells, regulatory T cells, some unidentified T cells as well. And if we look at the clusters more closely, we could find many naive T cells. The CD8 T cells with cytotoxic gene expression with differential levels of the exhausted molecular expression, which could be a target of immune checkpoint. And this immune cells also show the different proportion in the normal tissues and also tumor tissues. So there's a decrease in the NK cell proportions, increase in the regulatory T cells. We have not really tore apart the CD4, CD8s here. But in the next analysis, the CD8 cluster with more effector functions, there's more cells in the normal lung here, shown here. And for the exhausted T cell population, there's more in the tumor tissues. So using this type of analysis, we could identify different proportion of the different population in normal, tumor, and also in the metastatic site. And this is really the final piece of data. After looked at the different cell population, using our data, it's possible to look at the different interaction. It's actually inference of the interaction because we are looking at the transcriptome data. And by using a database, annotated the receptor ligand expression, we were able to generate an interaction map here. So compared to the normal lung, there's a role of a myofibroblast, which has many interaction with cancer cells, endothelial cells, and also T cells as well. So using our data set, looking at different cell population, we can look at the interaction of different populations and also correlation of different cell types. For example, here we have looked at the state 2 tumor cells, which we identified associated with tumor progression and metastasis. They have a correlation, let's say here, with the number of myofibroblast, CAF. So using this type of data, we can identify different signatures for different cell types and also their relationships, their role in tumor progression and metastasis. This project is still ongoing, so hopefully after finish analyzing the data, we may be able to find more biomarkers that's related to cancer progression, and that could be a target for therapy from the cancer cell part and also from the associated microenvironment part. So this study was supported by the post-genome project from the Korean government and has done in collaboration with Myungju An in Division of Hematology Oncology in Samsung Medical Center. Thank you.